It's Mrs. Vance again, and the last part of our notes for Unit 8, Part 4, is about mutations. You've heard the term mutation before. Now we're going to learn about what that means. So, a mutation is a change in the nucleotide sequence of DNA. And there are lots of ways that can happen. We'll talk a little bit about that as time goes on. But basically, mutations can involve big pieces of chromosomes or sometimes just a single nucleotide. If one nucleotide is wrong, then that can change the protein that's coded for. And so think about when you're typing something on the computer, how often do you make a mistake that you have to back up and correct or use spell check to correct? The cell has spell check also basically in the DNA polymerase. But sometimes mistakes still pass through. And so if those mistakes pass through, they're called a mutation, and that's going to be a change in the sequence. So there are two main categories of mutations within the gene. That's going to be a DNA mutation. Basically, we can have, first of all, base substitutions. If one nucleotide gets replaced with another, if you inserted an A instead of a G, okay, then that's going to change the sequence there. Sometimes these substitutions don't have any effect at all. They produce a silent mutation, sort of like two words that sound alike that have different spellings. Okay, so that may not have any effect at all, called a silent mutation. That, in other words, it's not going to change anything in the protein. It's just a difference there. Secondly, they can change the amino acid coding. So it could produce a different amino acid if you changed, one codon to another codon that happened to change to a different amino acid. That would be a different one. Okay, that's called a missense mutation. It changes one amino acid in the sequence of all those amino acids in that protein, and sometimes that can have a big effect on the protein. Sometimes it can lead. That can be sometimes an improved protein. Sometimes it gets better, or it could also change an amino acid into a stop codon. This is called a nonsense mutation. In other words, it stops before it's finished, and so you end up with an incomplete protein that is going to be non-functional because it's incomplete. Okay, so lots of things can happen. Just because it's a change doesn't mean it's bad, depending on, especially this silent mutation is not at all bad. Sometimes the missense mutation can actually improve the protein, sometimes not. It just depends on the situation. But we definitely are always going to have a big problem when we have a premature stop codon or a nonsense mutation. The other kind of mutation that we can have is a deletion or insertion. So maybe if you insert an extra letter in a word, I have a tendency to do that sometimes when I'm typing. I'll just stick an extra letter in there and have to go back and delete it, okay? Or if you delete a letter, okay, that's going to change the reading frame, the triplet grouping of the messenger RNA, so that the nucleotides are now not grouped into the same codons they were codoned before, and that can lead to big changes because basically everything from that point forward or downstream is going to be changed or may be changed, and that's probably going to produce a non-functional polypeptide, and that's not a good thing, because you needed that protein to do some specific job, and if it can't do it, then that's going to lead to problems in the organism. Okay, so here we have an example. Okay, normal hemoglobin DNA has a long sequence of bases in the DNA. This one particular spot has the codon CTT. We call it codons in DNA as well. The messenger RNA codon would be GAA, and normal hemoglobin that is made from that has the amino acid glutamine here in that particular spot. Sometimes the T gets substituted for an A in the DNA. That's going to now code GUA in the messenger RNA, and GUA codes for a different amino acid, valine, and that produces something called sickle cell anemia, which is a very bad condition. We'll talk more about it in class, especially when we talk about genetics coming up soon. But it definitely is very, very bad, and it's harmful to the person that has it. They have hemoglobin that's not shaped properly, that changes the shape of their red blood cells, can be very painful, and can eventually be deadly. So that's an example of a substitution that actually ends up being not a good thing. The other kinds that you have are going to be deletions and insertions. So let's look at some examples here of what might be different. Okay, so in a normal gene, Here's our normal gene that codes this particular sequence of amino acids. If we substitute in this particular spot here an A for a G, that changes that amino acid to serine. Okay, that might be harmful, it might be beneficial. We don't know until we see what the protein is. If you delete a nucleotide, if we take out this U here, everything is going to shift over to the left, basically, and so it's going to change the reading frame. So instead of coding for UUU, which is phenylalanine here, it codes UUG, which codes for leucine. And instead of AGC or GGC, we now have GCG, which codes for alanine instead of glycine. And then our next codon there would be shift off everything down, and that would be CAU instead of GCA, and that codes for histidine instead of alanine. So everything from that point forward is going to be changed or different. Same thing happens when we insert one. If we stick something in extra one in here in between the UU and then the third U, then that's going to change again. Everything from that point forward is going to be changed. Sometimes that can change one of these codons into a stop codon, which would even stop it before it even got finished. And so that would be that nonsense mutation. So mutations can be beneficial, they can be harmful, they can be neutral and not have any effect at all. It just depends on what the particular change happens to be. Now, we also mentioned that there could be chromosomal mutations. Sometimes those kind of things can happen in chromosomes during the process of mitosis or meiosis. And so if you delete a portion of a chromosome here, that's going to make a smaller chromosome. It's going to be not just one gene, but a whole bunch of genes. That's really going to be a big change there. Sometimes part of one chromosome will break off and reattach backwards, so that these two sections here are reversed. That's going to change the sequences that are coded for in those particular regions. A duplication would be if one part got copied twice, then you have a whole bunch of extra stuff there, which might be beneficial or might be harmful depending on what it is. And then sometimes you have something called a reciprocal translocation, where part of one non-homologous chromosome breaks off and attaches to a different one. And so you end up with two chromosomes that have parts of each other rather than parts of the same one. We'll talk more about these when we talk about genetics, and we'll talk about how this happens and what kind of problems can result from that. But those are these are always going to be problematic. They're going to cause big changes, big problems in the offspring. And that's the end of the notes for Unit 8.